Okay, we're going to move on to lecture 16, which is the last of our digestive system lectures. I spent a lot of time talking about the gastrointestinal tract, and that is the singular tube going from the mouth to the anus. And now we're going to look at some of our important accessory structures. So, structures that are important in digestion, but through which food does not pass. So, our first accessory structure we're going to look at is the pancreas. We did also talk about teeth, tongue, and salivary glands. Those are also accessory structures. But now we're moving down into the abdominal pelvic cavity. And the first major accessory structure we're going to look at is the pancreas. The pancreas is really important in digestion, but it's also a very important endocrine gland structure. We're going to focus just on the digestive aspect of it. The pancreas, the majority of it is exocrine in function, meaning that it makes secretions, which it then produces and releases into the small intestine. This will assist in the chemical digestion of essentially all of our organic molecules. So it helps to break down carbohydrates and proteins as well as fats, and also helps in the neutralization of acid chyme. So I have there, has an enzyme as well as a chemical secretion that it accomplishes. Location-wise, it sits essentially right on the medial side of the duodenum. Remember, the duodenum is sort of that C-shaped initial segment of the small intestine. So it sits in that medial concavity of the duodenum, and then will extend behind and slightly underneath the stomach, all the way across to the left side of the body, where it then essentially projects right at the spleen. It's about 20 centimeters long, so it's actually quite long, especially for being a gland. And again, it kind of moves all the way from the right side of your abdominal cavity across all the way over to the left side. So these are artistic renderings of the pancreas in the abdominal cavity. This one over here is isolated, and so you can see this esophagus coming down. That's the foot-long tube that runs through the thoracic cavity, goes through the diaphragm at the central tendon, and then becomes the stomach. The stomach, remember, is this sort of temporary storage structure for ingested substances. It begins the digestion of proteins, ending in the pylorus, and then the pylorus begins where, I should say, the pylorus ends where the duodenum begins. And so this is the C-shaped duodenum. Sitting in this medial border is the head of the pancreas, and that's going to go behind the stomach and slightly underneath it, all the way over across to the spleen. So this sort of rutted, dark red structure here is the spleen. And you can see the end or the tail of the pancreas actually is right against the spleen. If we look at this artistic rendering of the pancreas relative to the major digestive structures and abdominal structures, again, we're going to see that it goes all the way from the right side of the body over here as it's kind of tucked into that medial border of the duodenum, all the way across to the left side of the body. All right, the pancreas has three anatomical regions. You have a head, which is sort of this broad large portion that sits here on this medial concavity of the duodenum. It houses the exit point into the duodenum from the pancreas to the duodenum. So we'll see there's a large pancreatic duct that's going to exit into the duodenum, this first segment of the small intestines, through the head. And then we have an elongated portion we call the body, which houses the majority of the pancreatic duct. And then we're going to find that the pancreas sort of ends in this blunt portion here called the tail, which will be right adjacent to the spleen. So again, we have the head, which is sort of this broad, kind of flat portion to it. The body, which is this elongated, sort of narrow portion that traverses across the body cavity. And then we have the tail, which is this sort of blunt point that will be adjacent to the spleen on the left side of the body. Oh, and just looking at the texturing of it, it looks very, very similar to the salivary glands. Salivary glands are also exocrine, which means they're going to secrete outside themselves into a series of ducts that get carried into the GI tract. Here we find that also we have very, very tight lobation. It kind of looks like cauliflower. So you have these really tight sort of lumpy lobes, and coloration-wise, it's sort of a ivory color, sort of a little bit darker ivory color. All right, the function, the majority of the pancreas is exocrine. So you have about 99% of what it's doing is secreting what we call pancreatic juice, which is a combination of acids and enzymes, as well, sorry, not acids, enzymes and bicarbonate, which will neutralize the acids. So we're going to see that sodium bicarbonate is a really important secretion produced by the pancreas. The sodium bicarb functions to neutralize the acidic chyme coming out of the stomach. So if you remember, the stomach has a pH of somewhere between 1 and 2. That's going to facilitate protein digestion, and it's going to activate pepsin in the stomach. But when that acidic chyme comes into the duodenum, it has to be immediately neutralized. So even though the duodenum is a very short segment of only about 10 inches, we're going to find that by the time you get to the end of the duodenum, the pH of the chyme has gone up to about 8, which is moving it from a very acidic 1 to 2 pH up to not just neutral, but a little bit basic. So that's really important so that we don't corrode and damage the inside of the small intestine. In addition to the sodium bicarbonate secreted by the pancreas, we also find a suite of enzymes that essentially continues the breakdown of the carbohydrates and the proteins and initiates the breakdown of lipids. So carbohydrates begin their digestion in the oral cavity with salivary amylase. Proteins begin their digestion in the stomach with pepsin. And both of the carbohydrates and proteins at this point, as they arrive in duodenum, still haven't been broken down to absorbable units. So we're going to continue to help facilitate the digestion of carbohydrates and proteins with the pancreatic secretion. We're also going to initiate the digestion of lipids. So lipid digestion is done by lipases, which are secreted by the pancreas and also into the small intestine of the duodenum. So we'd say 99% or almost all of the functional cells of the pancreas are exocrine and involved in making the sodium bicarbonate and the enzymes. There is a very important endocrine function that the pancreas will also carry out. We'll spend a little bit of time looking at when we deal with the endocrine system. But the important and notable hormones secreted by the pancreas are insulin and glucagon and somatostatin. Insulin and glucagon are really important for nutrient regulation, secreted by beta cells and alpha cells. And then somatostatin is a growth hormone secreted by our delta cells. Okay, so if the majority of what the pancreas is doing is secreting, I should say releasing and secreting exocrine substances into the small intestine, those secretions have to be carried and delivered to the small intestines by ducts. And for the pancreas, there are actually two major duct systems. The primary duct system is called the pancreatic duct, and then it has a smaller accessory duct. So in this drawing, you can see traversing from the tail through the majority of the body and then down to the head is the primary or main pancreatic duct. And then a little bit superior to that is the accessory duct. What's really important about the pancreatic duct, the main one, is that as it comes into the lumen of the small intestines, it will actually unite with secretions coming from the gallbladder and liver. So this actually will be bile, which is why the structure looks green. Bile and pancreatic juice enter into the small intestines through this common aperture or opening. And this is referred to as the hepatopancreatic ampulla, which is the chamber. And then you have a little sphincter here of smooth muscle that helps to regulate the movement of substances through the pancreatic duct and the bile structure here called the common hepatic, or sorry, the common bile duct. And this is called the sphincter of Oddi, or the hepatopancreatic sphincter. All right, so two main ducts, like I mentioned, we have the pancreatic duct, which is going to be the main delivery duct for pancreatic juice into the duodenum. Again, it's going to unite with the common bile duct coming from the liver and gallbladder carrying bile. And as those two ducts merge together, they form a central chamber, common chamber, called the hepatopancreatic ampulla or duodenal ampulla. So in this drawing here, the ampulla is this sort of enlarged area here where we're going to see sort of this common passage for both bile through the common bile duct and pancreatic juice through the pancreatic duct. So another name for this is duodenal ampulla. So it's this chamber that exists within the duodenum. I actually prefer hepatopancreatic because it reminds me that it's carrying substances from the liver, which was where we get this term hepat. Hepat means liver, and pancreas. All right, then we also have a smaller accessory duct. So this is the smaller accessory duct and just allows for a little bit further movement of pancreatic juice. So you can release a larger volume of pancreatic juice at one point or one time. So it's smaller and it's a little bit superior to the main pancreatic duct. All right, so this is a video actually you saw before. I'm just gonna play the beginning of it. This is where we're traveling down through the esophagus. So you can see a nice peristaltic wave produced by the muscular cisterna. That's the lower esophageal sphincter. And now we're moving into the stomach where I can see rugae. You're going to look up toward the fundus. We'll see some animation where ingested substances are falling into the stomach. Another wave of peristalsis as we then move these substances down toward the pylorus. And then you can see the small aperture or hole from the pylorus. And looking through that, we're now into the small intestine. So notice the plicae circularis. And as I move in, this is a very short segment. So we almost immediately come upon the accessory duct, which is right there, and then the main pancreatic duct. So that was really fast. I'm going to rewind this a little bit so we can see again. We're coming up in this first duct that's going to sit right here. That's the accessory duct right there. That's pancreatic juice. And then that's that combination of bile and pancreatic juice coming from the hepatopancreatic ampulla. Okay, and now we're just going to continue down through the small intestine. All right, so histologically, the pancreas is glandular. I mean, that's its job. It's exocrine primarily and endocrine, but it's just a huge gland. The gland is lobulated, meaning that it has distinct sort of aggregations of exocrine cells. The exocrine cells that make up the majority of the pancreas are referred to as acinar cells, and they're the ones that are going to be making the enzymes and the sodium bicarb. All right, and then here you can kind of see this is what we call an acinus. This is sort of this sort of sac-like structure here. So this is an acinus, and here's another acinus. And so the cells that make up this acinus are called acinar cells. Notice that in this simplified drawing, yellow are the exocrine acinar cells, and you can see the blue arrows representing sort of the pathway that pancreatic juice would follow from the acinar cells out through these smaller ducts into the main traffic of the pancreatic duct, through the hepatopancreatic ampulla and the sphincter, and then into the small intestines at the duodenum. All right, so those are acinar cells. When we look at the endocrine function, the endocrine cells make up by number a very small amount of functional cells within the pancreas. They make up less than 1% of the pancreatic cells, but their influence is all over the body. The pancreatic cells secrete hormones from these sort of aggregations of endocrine cells. And these are called islets of Langerhans, or islands of Langerhans, sitting in a sea of acinar cells. So right here, you can kind of see these lighter cells here. These are very cuboidal. It has a nuclei right in the center and sort of this light cytoplasm. So these are the endocrine cells. We call this an islet of Langerhans. And they're the ones that are going to be making insulin that functions to allow blood sugar to go from blood into cells. And then glucagon, which does just the opposite. Glucagon allows you to release sugar into the blood. These two hormones not only move sugar in and out of cells and in and out of blood, but also all of our nutrients, so amino acids as well as fatty acids, are going to be influenced by insulin and glucagon. And then lastly, somatostatin, which is just a growth hormone. So I have here insulin and glucagon help regulate blood sugar. So when your blood sugar levels are high after a meal, you release insulin, and that insulin allows you to uptake and utilize nutrients, specifically sugar. And in between meals, where your blood sugar levels may be dropping, you're going to find that glucagon is going to be responsible for releasing nutrients, specifically glucose, into the blood. All right, so this is again another drawing of the pancreas. What they're trying to draw in blue here are the islets of Langerhans that kind of sit scattered throughout the pancreas from head to body to tail. And then you have aggregated around the duct system all these acinar cells. So this is just a nice sort of isolated view of one of those islets of Langerhans sitting in a sort of sea of floating acinar cells making pancreatic juice. This is a nice magnified view of the histology of the pancreas. So when you look at a histological image of the pancreas, how can you tell that's what you're looking at? Well, first of all, you're going to see acinar cells aggregated in sort of circular patterns. These cells are cuboidal to kind of columnar. So when you look, they basically have their nuclei around a periphery of a sort of circularly arranged acinar cells. In the center would be where the duct is. So the nuclei are at the base, and then the cells kind of project toward the center. We're going to see that arrangement again and again and again, where they're kind of like in a round circular structure. The acinar cells tend to stain a little bit darker. Their cytoplasm tends to be a little bit darker and more solid. The nuclei tend to be at the base of the acinar cells, and they're typically arranged into rings. And when we look at the islets of Langerhans, these are going to be much lighter in the sense of the cytoplasm staining, and the cells are going to have nice round nuclei kind of sitting in the center. So you see this sort of aggregate or island of islet cells sitting in a sea of acinar cells. And so that's what we want to look for when we're looking at a histological image of the pancreas. All right, next we want to look at the liver. Anytime we hear liver, we're going to think hepat, because hepat means liver. So anything referring to liver will have that prefix of hepat. So we have the hepatocytes, which are liver cells. We have hepatic ducts. We have hepatic vessels. So anything dealing with the liver is going to start off with hepat. The liver by far is one of the largest abdominal organs. It weighs about 3.5 pounds in 70 kilogram male. It sits on the right side of the abdominal pelvic cavity, sort of in that superior right quadrant. So when you're looking, you're going to see this very solid mass of functional cells that sits predominantly on the right side, tucked underneath the diaphragm. So this is the diaphragm right here. The liver is very dome-shaped. It sits underneath the sort of curvature of the diaphragm. It's bisected unequally into a right and a left liver lobe. The bisection is done by the falciform ligament, which we talked a little bit about in lab. The falciform ligament is simply a mesentery, but it's filled with a lot of collagen fibers, so it's very supportive and thick and not easily stretched. And that's important because this is the structure that's going to help, along with the central tendon, to literally adhere or stick or hold the liver underneath the diaphragm. So every time you take a breath, when you're breathing, when you take a nice big inhalation, that is facilitated by the diaphragm lowering and essentially the liver kind of helping to weigh that diaphragm down. And when you take an exhalation, we're going to see that the diaphragm becomes more dome shaped and the liver is going to be moving up. So the liver literally moves up and down with every breath, since it literally is stuck to the undersurface of the diaphragm itself. So this is showing you the diaphragm right here. And then the falciform, remember, is right down the midline. So if you were looking right about this location, you would find, the sternum, and then right behind the sternum would be the heart. So it's sitting right in the center of the abdominal cavity. And then we can see a little bit of the gallbladder here underneath the right liver lobe. And then this is the round ligament, which is a vestigial structure. It's left over from the umbilical vein that would have traveled from the individual's umbilicus when they were in the fetus of their mother, and then traveling past the liver toward the heart. Okay. So metabolic functions. Actually, I should say the liver has many, many, many functions. One of the most important is its metabolic functioning. When you look at the blood flow to the liver, we're going to find there's two major sources of blood that are going into the liver. One is from the digestive tract. So when you look at the GI tract, and that means stomach, small intestines, large intestines. And again, remember, you're looking at about 20 feet of small intestines, another 5 feet of large intestines, another, distance-wise, about not quite a foot in the stomach. You're looking at quite a distance of small intestine, large intestine, stomach that's going to be absorbing nutrients into blood. All of the nutrients that arrive in that absorbed blood are going to travel not to the heart directly, but through the liver and then to the heart. So all of the blood leaving the digestive tract is going to first travel through the liver. The liver's job is going to be to metabolize all those raw nutrients. It'll take the huge load of raw nutrients you just have absorbed after eating a meal, and it's going to need to do things like detoxify any of the substances coming in. It's going to function to initiate processing of nutrients coming through, like the nutrients that arrive through the lymphatic system initially for lipids. It will store and metabolize carbohydrates as well as proteins. So all of the blood, and that's what this drawing here is trying to show you, all of the blood that is leaving the digestive tract, carrying all that wonderfully absorbed nutrient and water, is going to travel first through the liver before it ever reaches the heart to be circulated out to all your other organs and tissues. Now, the vessel that travels between the GI tract here and the liver is referred to as a portal vessel. A portal vessel is any vessel that sits between two organs that's not the heart. So in this case, we're moving between the GI tract and the liver. And so this is what we refer to as the hepatic portal vein, because it's a portal vessel sitting between the GI tract and the liver, and specifically it's going into the liver. So we refer to it as the hepatic portal vein. We'll talk more about that also in just a minute. So the function is to take all these raw nutrients that you've just absorbed and then start to metabolize them, store them, repurpose them, detoxify any of the nutrients coming in. So the cells that do this major job are called hepatocytes. Again, hepat means liver, cyte means cell, and this makes up the majority of what are liver cells. About 60% of the cells in the liver are hepatocytes. Again, their job is to monitor and adjust blood nutrients. They're going to help to store and remove excess nutrients. So, for example, one of the primary places that we store carbohydrate is in the liver. And based on the size of the liver, it actually stores the greatest concentration of carbohydrate as opposed to any other organ system. So the skeletal muscle is the other organ system that stores the majority of our glycogen. They estimate that about 8% of liver weight is actually glycogen, which is the main storage of carbohydrate in our bodies. And then also the hepatocytes serve a function of detoxifying. Not that you need to know this, but I put a little drawing here depicting sort of many of the metabolic pathways that occur within a common hepatocyte. So all of these arrows are showing you biochemical pathways that are utilized by hepatocytes. So just to see the complexity of metabolism that takes place. All right, in addition to the metabolic function, the liver has a very important hematological function. He means blood, so this is a function that the liver has in the sense of moderating blood constituents. So first is the liver houses what we call Kupffer cells. Kupffer cells are more like immune cells. They're phagocytic, which means they eat other cells and eat cellular debris. Phagos means to eat or consume. So these are big eating cells. And they're going to sit in the spaces that blood would be flowing through the liver. And their job is to remove old and damaged blood cells, cell debris, and pathogens. A pathogen is any disease-causing agent. So that may be a bacteria, it could be a fungus, or it could be potential viral infections that are traveling in the blood through the body. One of the other hematological functions of the liver is to release blood proteins. The majority of blood proteins are produced by the liver. Some of these are going to be our blood clotting factors, blood proteins that regulate our water content, so that would be the osmotic pressure, and then also carrier proteins, proteins that will help carry our sort of water soluble vitamins as well as steroids. All right, and then one of the last functions, although this is a very short list, is the bile synthesis and secretion. So bile is actually a waste product, but it's a very useful one, and so we use it in conjunction with digestion. Bile is simply a breakdown product of hemoglobin. Blood cells circulate in blood for about 120 days, and when those blood cells are starting to destabilize, mature red blood cells do not have a nuclei, and so they don't have a capacity to repair anymore. So eventually they start to break down. It takes about 120 days. And when that starts to happen, those cells are called senescent. So they're starting to go through this process of getting old and breaking down. We have cells throughout our body that are purposed in breaking down those red blood cells. One of the major locations that red blood cell turnover takes place is in the liver. Another place would be in the spleen. So the liver's hepatocytes are going to start to break down the pigmented component of hemoglobin, and they produce a product called bilirubin. Bilirubin is actually quite basic, and it also is amphipathic, meaning that it has a charged area to it and an uncharged area to it. And because of that, it can surround fat and oils, and it helps to break them into smaller sort of lipid droplets, very similar to soap. So we're going to find that bile, which is a waste product from normal red blood cell turnover, is going to be used to facilitate the neutralization of chyme. So because bile is very basic, we're going to help to neutralize that chyme as it unites with the pancreatic duct and the sodium bicarbonate. And then it also helps to emulsify. One of the hardest aspects of sort of fat and oil digestion is that our body's pretty warm, and so fats and oils melt. And most of our secretions are water-based, so you know that water and oil don't mix, and so you basically get these really big mats of oil, and enzymes are supposed to get in and break bonds. The problem is that if it's a big lipid droplet, there's not a whole lot of surface area to facilitate access of the enzymes to those bonds. And so what the bile does is it breaks those big lipid droplets into small lipid droplets, just the same way soap does when you're washing dishes. And that then allows enzymes to get in and break up all of those bonds utilizing the lipase enzymes. Now, we make bile all the time, so we're constantly breaking down red cells, so we're constantly producing bile. But we aren't always releasing bile. Bile is excreted or gotten rid of through the digestive tract because of the fact that it facilitates lipid digestion and acid neutralization. So if we make it all the time, but we're not releasing it all the time, we have to have some place where we store bile. And that storage location is called the gallbladder. And that's located just underneath the right liver lobe, which we can see right here. The gallbladder and bile structures, which are called biliary structures, are typically green because bile itself has that green pigmentation to it. So I'm very similar to if you've ever seen a bruise healing. So if you ever get hit and you break some blood vessels underneath the skin, the blood will kind of ooze out of the vessels, and initially it'll be red where the injury occurred. And then as the oxygen on the hemoglobin starts to dissipate, it becomes more and more blue. And so when you see a bruise, as the healing is occurring, you're going to see it go from red to kind of blue. You get that kind of black and blue appearance to it. And then it starts to get green and yellow. And that green and yellow is where we're producing the bilirubin. So we're breaking down that hemoglobin in the tissue. So take-home message is that bile is green. All right, so I want to talk a teeny bit about gallbladder stones and show you a little video. But when we store bile in the gallbladder, the bile can start to get too thick for a variety of reasons. One of the main reasons is there might be too much cholesterol in it. And it gets really sticky and it forms what we call gallstones. When those gallstones are normally formed, for most of us, we get tiny ones that we pass all the time. So those may be the size of grains of sand or little seeds that we can pass through those bile structures or biliary structures. But sometimes they can get large enough that they get stuck. And that's where we have problems with gallstones. So this little video is going to talk a little bit about them and some of the risk factors and what they do with gallstones. All right.
It's something you rarely hear about, but 10 to 15% of Americans suffer from gallstones.
Gallstones are a combination of things, actually. They form because they form from bile, and bile contains several things, including cholesterol, calcium, and also pigment.
The stones form in the gallbladder, a small pouch beneath the liver that stores the bile we need to help break down food.
And oftentimes, if there's an excess of cholesterol in the bile, then it can become supersaturated and actually form stones.
Christy says the majority of stones pass right through our systems and never cause problems. But when they're big enough to block a bile duct itself, it can be painful.
That pain can be very sudden and severe and very sharp. Typically, it does occur after eating, but it can occur at any time.
The pain is most often felt in the right upper part of your abdomen, and sometimes right in the center, just under your breastbone. But Christy says there are several ways to prevent gallstones.
We know people who are overweight or obese have a higher prevalence of gallstones. We also know that women have a higher incidence of gallstones compared to men. But to lower your risk of developing gallstones, obviously you want to eat a diet that is high in vegetable proteins and nuts. Also, maintaining a healthy weight is important as well, and exercise.
Genetics can play a role too.
We know that people who are of a Hispanic origin, specifically Mexican Americans and also Native Americans, have a higher prevalence of gallstone disease.
And pregnancy.
And this is due to estrogen and progesterone that causes an increased saturation of cholesterol in the bile.
So if you fall into one of these ethnic groups, or if you're pregnant, you should be extra vigilant.
A person should see their doctor if they're having symptoms of gallstone disease, including that right upper quadrant pain that may be sharp, and it's not going away.
Treatment for symptomatic gallstones often means surgically removing the stone or the gallbladder itself. But both procedures are done with small, minimally invasive endoscopes. And Christy says, not to worry.
Most people do fine without the gallbladder in.
For Accent Health, I'm Christy Paul.
Okay, so again, kind of take-home messages there. Obviously, if you're at higher risk, you want to be aware. So that would be females, Mexican Americans, Native Americans, anyone that has excess weight. So if you have a condition of diabetes that can cause excess weight gain, or sedentary and not exercising may have excess weight gain, those all put you at higher risk. So if you have upper right-hand quadrant pain, sometimes just simply ongoing nausea, vomiting, those are all signs that you may have gallstones. All right, so next thing, this is just a really nice table that shows all the different functions that the liver carries out. And again, this is still even a short list of what the liver does. But notice in yellow here, we have metabolize and or break down nutrients from food that we have ingested into energy when it's needed. So this is a really important aspect of what the liver is doing. So remember, all of the blood coming from the digestive tract will have to travel through the liver before it's sent out to all of our other organs and tissues. Part of this is going to be up here, you can see in this top corner, to remove potentially toxic byproducts. We have an immune function in the sense that we have cells that are going to function to fight off bacterial infections. Those are our Kupffer cells, initiating infections, or initiating the, I should say, the, not the infection, but the immune system's recognition of an infection. We also have here produces most of the substances involved in blood clotting, because we're going to find that most of the blood proteins are produced by the liver. We also know that the liver will be tasked with breaking down hemoglobin. So in the process of that, making bile. So I have here producing bile. Bile is really important because it helps to digest fat and also helps to absorb the water-insoluble vitamins that we have in ingestion. So vitamin A, D, and K. These are all lipid soluble, so it requires that we have bile in order to absorb them. And then I have here prevents shortages of nutrients, and one of the ways it does that is it stores nutrients, vitamins, and minerals until periods of time that we need them. So many, many important liver functions there. All right, so just a little bit of the gross anatomy. When we look at the liver, again, it's a very large solid organ that sits in the upper right-hand quadrant of our body. It's divided actually to 4 lobes, but we really only recognize 2. So there's a very large right liver lobe, which we see over here. It's dome-shaped because it sits actually under the curvature of the diaphragm. Sometimes you'll see sort of contours or ribs through it. The right liver lobe is separated from the left liver lobe by the falciform ligament that actually runs right up and down the body's midline. And then the left liver lobe obviously is sitting on the left side of that falciform ligament. We have 2 smaller lobes called the caudate and the quadrate lobes. These guys are kind of more like little lumps on the undersurface. But the caudate lobe, which we have here, this is actually looking at the undersurface of the liver. So if you're looking under the liver, the posterior aspect of it, so this is the side that's going to be against your back and your vertebrae. This is the caudate lobe, because caudate or caudal means tail. This is the front of the liver, and this is where we have the quadrate, which means the four-sided lobe, so quadrate. And then we can see the gallbladder also sitting underneath the right liver lobe here. So the caudate and the quadrate, the right and left liver lobes actually don't do anything differently from one another. They basically do the same job. And so physiologically, it doesn't really make much difference what lobe we're looking at. In fact, if you were to do a liver transplant from a large person into two smaller individuals, which they often will do since livers are fairly scarce in organ transplant, you can actually split a liver, and it doesn't really matter what part you split because all parts essentially do the same thing. All right, blood circulation is really, really, really important in the sense of what's happening in the liver. You'll notice we have in this drawing the liver right in the center, and we have two inputs of blood. So we have two sources of blood, so blood going in and in, two locations into liver, and one output. So two inputs, one output. And what we're going to find is that one of the inputs is coming through this hepatic portal vein. And this is the blood coming from our gut, right? The blood coming from your GI tract. So this is what we refer to as workload blood. So blood coming from the gut has raw nutrients, nutrients that haven't yet been metabolized or balanced or regulated. They haven't been detoxified. They haven't been stored or handled. So this is just pure nutrient that gets absorbed. The GI tract doesn't regulate, for the most part, how much sugar you absorb or how much protein you absorb or how much lipid you absorb. Its job is really just absorb. So if we eat too much sugar, the digestive tract doesn't manage that. It just absorbs all of it. So we have all this raw nutrient that's coming into our bodies. And this raw nutrient has to first pass through the gatekeeper of the liver before it travels out into our systemic circulation through the heart. Now, the liver is very metabolically active. About 8 to 10% of our metabolic energy is used by the liver. And so we need a high volume of blood flow carrying oxygen-rich blood from the heart to travel directly to the liver to help support that very high metabolic function. We estimate 20-25% of our cardiac output is going to be moving through the liver. So that means every time your heart beats, a fifth to a quarter of every heartbeat's blood volume is going to be traveling directly to the liver to help support those hepatocyte needs. So this is the hepatic artery. We can see here oxygen-rich blood coming into the liver on the hepatic artery. And then we see the oxygen poor but raw nutrient-rich blood, which is this workload blood, also traveling into the liver. So here's the workload, here's the sort of oxygen to allow for the energy conversion. And then once blood has been metabolized and balanced, detoxified and stored, all the things we need, release what we also need circulating, that processed blood is then released through the hepatic vein. And then the hepatic vein will merge into this vessel called the infravena cava that travels directly to the heart. So I have here, there's two inputs of blood. We have the hepatic artery, which is going to be carrying blood from the heart, which will have just perfused through the lung, increasing the levels of oxygenation to somewhere around 98-99%. So this very richly oxygenated blood will then travel to the liver. Again, upwards of a fifth to a quarter of our cardiac output, upwards of 25% of your cardiac output is going to be traveling directly through the liver. And then we have our workload blood. This is blood coming from our GI tract through what we call the hepatic portal vein. So this is blood that's going to be low in oxygen because it would have just gone through all of the GI tract, which would have used the oxygen, but raw nutrient dense as well. So we're going to see we have this sort of raw nutrient workload blood that needs to be processed by the liver entering through the hepatic portal vein. So this is just a nice artistic drawing. Here we see the liver, and we're showing the stomach, the duodenum, jejunum and ileum here, and a portion of the large intestine. You can see these blood vessels here that are essentially receiving nutrients from the GI tract and carrying them toward the liver. So these vessels here are called the mesenteric vessels because they're in the mesentery of the small intestine. And they will then drain into the hepatic portal vein that also receives blood from the gastric and duodenal vessels. These will then go into the liver to be processed, to be detoxified, and to be balanced. So the metabolic function of the liver will be to process that raw nutrient-rich blood coming in from this hepatic portal vein. Because of the high level of metabolism and energy that takes place in order to do all those metabolic functions, we also need to have oxygen-rich blood coming in through the hepatic artery, which we have right here. So this will be carrying oxygen-rich blood to help support the metabolism of all the hepatocytes to do their job of processing this raw nutrient-rich blood coming in from the GI tract. All right, so this is a really nice artistic rendering of blood flow circulation. So here they have the liver over here. So there's the liver. And notice you have two inputs of blood. One is blood coming from the hepatic artery, carrying this wonderfully oxygenated blood. And then we have blood coming in from the hepatic portal vein from the GI tract and some of the surrounding tissues like the spleen. So this is carrying raw nutrient into the liver. The liver will process all the blood traveling into it, metabolize, regulate, detoxify, and then that processed blood will exit the liver through the hepatic veins and then eventually merge into the vena cava and then go out to the heart to then get sort of distributed throughout the body. All right, so the hepatic vein is going to be the single output from the liver, and that's going to be all that wonderfully processed blood. All right, so I'm going to show you this quick video looking at liver structure. This is really good. It goes into a little bit more detail than we'll need, but it does a very good job in showing you the liver structuring.
The liver is an important as well as structurally and functionally complex organ. One of the important functions of the liver is to receive and take up nutrients and other molecules absorbed from the intestines before the blood is returned to the heart via the inferior vena cava. The liver has dual blood supplies, one venous and one arterial. The portal vein, which carries blood from the spleen, pancreas, and intestines, contributes about 75% of the blood volume to the liver. This blood is rich in nutrients and other absorbed molecules, but relatively poor in oxygen content. The hepatic artery, a branch of the celiac trunk from the abdominal aorta, contributes about 25% of blood volume entering the liver. This blood is well oxygenated, but nutrient poor. Blood from the portal vein and hepatic artery mixes as it enters and passes through a liver lobule, the classical structural unit of the liver. Blood then percolates through the lobule and is collected by the hepatic or central vein. Central veins unite to form larger sublobular veins, which ultimately join the hepatic veins. Blood flows to the heart via the inferior vena cava. Bile comprises the exocrine secretory product of the liver. It is stored in the gallbladder. During digestion, bile is released from the gallbladder through the cystic duct into the duodenum to aid in the digestion of fats. The filtration of blood and the production of bile takes place within the structural and functional unit of the liver, the lobule, where the terminal branches of the vessels carrying incoming and outgoing blood meet. The liver lobule is roughly hexagonal in cross-section. In the center of the lobule lies the central vein, which collects blood from the lobule and carries it to the hepatic veins. Located at the lobule periphery are branches of the vessels supplying incoming blood, the portal vein and the hepatic artery. Also located here is the bile duct. Collectively, the portal vein, hepatic artery, and the bile duct together comprise the portal triad. Taking a horizontal cross-section of the lobule gives us an arrangement typically seen in histological preparations. In this view, we can identify the central veins and associated portal triads. There are two prevailing views of liver lobule organization: the classic liver lobule, or anatomical model, and the liver acinus. The classic liver lobule is based on the structure of the lobule we just discussed. It is hexagonal in shape and is defined by portal triads surrounding a single central vein. To identify a lobule in a histological slide, first find a central vein. Next, locate the portal triad surrounding that vein, usually about 6 at the corners of the hexagonal prism. An imaginary line connecting the triads represents the outside border of the lobule. In the classic liver lobule, blood flows from the portal triads toward the central vein. Bile flows in the opposite direction, from the central position toward the portal triads at the lobule periphery. Remember, the classic liver lobule model emphasizes the anatomical structure of the lobule. The liver acinus is another way to view liver structure. This model is important for understanding liver physiology and pathology. The structure of the acinus overlaps two classic liver lobules. The acinus is diamond or oval shaped. It involves approximately 2 portal triads and 2 central veins. To identify the acinus, find 2 adjacent portal triads. This makes the short axis. Next, find the closest central vein on either side of the triads. This is the long axis. The resulting shape is a diamond or oval. Blood still flows from the portal triad to the central vein, but within the shape of the acinus, the flow is from the center to the periphery. Bile flows in the opposite direction, from the periphery towards the center. The acinus model is important because it emphasizes the metabolic differences in hepatocytes between the portal triads and the central vein. The cells in the acinus are divided into 3 zones and are numbered in the direction of blood flow, from 1 to 3. Cells in zone 1 lie along the short axis, between the portal triads. Thus, these cells are first to receive everything in blood, including oxygen, absorb nutrients, and absorb xenobiotics. Cells in zone 2 have characteristics intermediate between the cells of zones 1 and 3. Cells in zone 3 are closest to the central vein, and therefore are the last to receive incoming blood. As blood moves towards the central vein, it becomes depleted of oxygen, nutrients, and xenobiotics. To review, the liver lobule is the anatomical model. It is hexagonal in shape. It involves 1 anatomical liver lobule, and blood flows from the periphery to the center. The liver acinus focuses on function. It is diamond or oval shaped. It involves 2 adjacent anatomical liver lobules. The blood flows from the middle, which we discussed as a short axis, to the periphery.
Okay, so now we're going to look at the hepatic liver lobule structure. So when we look at the hepatocytes, they're going to be arranged into columns around a central vein. So every lobule, which is this repeating hexagonal unit or structure, is going to have the same arrangement. So we're going to see a central vein in the center, and it's kind of like a funnel. You know, when you use a funnel for making I don't know, juice or something like that. So you maybe pour the fluid and it all collects in the central vein or the central portion of the funnel, and then you can drain the fluid from that funnel. So the exit point from a lobule is going to be through the central vein. That's the exit point. The entry point for fluid into the liver lobules is going to be at the periphery. So like in that little video showed, at the periphery at these corners, and you have, you know, a 6-sided structure, so you're going to typically see 6 corners, is where you're going to have the entry point for blood. So you'll notice there's a blue and a red vessel here. Obviously the blue one is the hepatic portal vein. The red one's the hepatic artery, because the red is oxygenated. So we have red coloration to indicate the artery and blue to indicate the vein. So blood is going to enter in at the periphery, and then it's going to travel between these columns of hepatocytes toward that central vein. So if we look at this little small set of liver lobules, you see how they fit together kind of like tiles in a bathroom. And the center of each of the lobules is that central vein. So each of the lobules is hexagonal. They kind of look like spokes on a wheel. So if you think about the central veins as the center to a wheel, for example, on a bike, you'd have the tire around the outside edge, and the hepatocytes are going to be in these columns that radiate out toward the tire. So these columns of hepatocytes are kind of like the spokes on a wheel. So we see that arrangement again and again and again. So these are 3 liver lobules associated with each other. And that's the classic sort of unit that we see repeating again and again and again within the liver. The estimate we have over 100,000 liver lobules per liver. All right, so when we look at a little bit closer view at a single liver lobule, we're going to look at 3 columns of hepatocytes and the associated central vein and incoming blood coming from the hepatic portal and the hepatic artery. So we have the central vein over here on the right. We have on the left here the hepatic portal and the hepatic artery. The arrows indicate the direction of flow. So blood's going to come from this periphery, flowing down through these spaces between the columns in the modified blood vessels. So these are called sinusoids. They're going to be lined with endothelial cells, which line all blood vessels and in the interior of the heart. So these are just extensions of those little blood vessels. These are called sinusoids. And you can see the blood will flow down through these, almost like little channels, from the periphery toward the center. And the idea is we're going to pass the blood past these sort of working hepatocytes who will be charged with metabolizing and regulating what is being carried in that blood. So carries blood to be processed past these columns of hepatocytes. Almost like you have, workers in a line here, and you have a conveyor belt that's traveling past them. And each of these hepatocytes will be charged with managing blood constituents. All right, so this is a nice artistic rendering of a single liver lobule. And they're trying to show you the arrangement of the hepatocytes in these nice columns. And then you can see we have at the corners, or actually the 6 edges of the hepatocyte, we see this sort of triplicate structure where we have the hepatic portal vein carrying our blood with raw nutrients we just absorbed, blood carrying oxygenated hemoglobin for all of the energy our lobule cells are going to need, hepatocytes. And so these will be the 2 vessels that are coming in. Notice here in this drawing, we have the arrows indicating the direction of blood flow. So they'll travel into the lobules, into what we call the sinusoids. The sinusoids will pass all the hepatocytes on their way to the central vein. And then the last structure we're going to look at is this one in green, which is the common bile duct. The bile canaliculi, which will eventually emerge into what we call the bile duct. All right, so this is actually out of your textbook, and this is a nice one because they're showing you the liver lobule. In the center is the central vein. You can see at these different apices or corners, we're going to see the hepatic portal vein and hepatic artery that deliver blood into the lobule from the periphery. That blood will then travel past all these columns of hepatocytes. Hepatocytes will process that blood on the way to the central vein. All right, this is another really great video looking at a structure which is not yet really well incorporated into most textbooks because it's relatively new. And this is really looking at the space between the sinusoid, which is the modified blood vessel, and the columns of hepatocytes. And so those are called the parasinusoidal space, or the space of Dicey. And then looking at the small little passages for bile that's being produced, and the sinusoid itself. So I'll play this one.
These are the most common cells present in the liver. Hepatocytes are arranged in the form of anastomosing plates. The cells radiate from the central vein towards the periphery of the lobule. The spaces in between the adjacent rows of hepatocytes are sinusoids. Thus, each liver cell is surrounded by sinusoids on two sides. These sinusoids are lined by an endothelium, which is fenestrated in nature. Surface of liver cell is separated from this endothelial lining of the sinusoid by space of Dissay. Each hepatocyte is polyhedral in shape. Surface of hepatocyte shows three types of specialization. Sinusoidal surface. Cell surface of hepatocyte adjoining sinusoids bear microvilli that project into space of Dissay. Canalicular surface. Bile secreted by liver cells is poured into bile canaliculi. These bile canaliculi are spaces present between the cell membrane of adjacent hepatocytes. The canalicular surface of hepatocytes shows longitudinal depressions, which forms the wall of a bile canaliculus. Irregular microvilli project into the canaliculus. Intercellular surface. These surfaces help in uniting adjacent hepatocytes. It consists of communicating junctions, which helps in exchange of substances between adjacent cells. Hepatocytes are highly active cells. Each hepatocyte contains numerous mitochondria. Abundant rough and smooth endoplasmic reticulum. Golgi complex. Lysosomes. Free ribosomes. And vacuoles containing enzymes. The liver cells also contain stored glycogen, lipid, and iron. Some hepatocytes are binucleated. In addition to hepatocyte, which forms more than 60% of cellular population, liver contains other cells like itocells, Kupffer cells, hepatic stem cells, lymphocytes, and connective tissue cells.
Okay, so this is relatively new histology, and it's really important in the sense of the pathology that results in the liver. So what I want to kind of bring your attention to is when we look at a column of hepatocytes, so here we have two columns of hepatocytes, running between them is going to be the sinusoid. And the sinusoid is just really a modified blood vessel that has relatively large gaps and spaces between the endothelial cells. So blood, as it's traveling through here with all the raw nutrient and components that need to be detoxified, they will then be able to escape from this relatively rapidly moving sort of flow of blood into a slower sort of more stagnant space called the perisinusoidal space. So this is what the video was referring to as the space of Dissay. The space of Dissay. And what's significant about the space of Dissay is it allows for much slower migration of fluid past the hepatocytes. So giving hepatocytes really a good chance of monitoring and regulating what is actually in the plasma. It's kind of like a really fast-flowing stream. If you've ever seen a fast-flowing stream, the water flows faster in the center, and then as you get toward the edges where the shore is, the water moves more slowly there. And so that's kind of the analogy. We want the fluid to move relatively slowly past the hepatocytes, so the hepatocytes have a chance to really well regulate what's in that fluid. Stop. Stop. All right. So this is what we call the space of Dicey, or parasinusoidal space. Peri meaning around the sinusoid. So I have here space between the hepatocytes and the sinusoid endothelial cells. It contains blood plasma. So plasma is just the fluid part of blood. So blood cells will remain within the sinusoid, and then the fluid part with the dissolved nutrients will be able to then be filtered out into this parasinusoidal space. And that slows down the movement, so that helps to facilitate the exchange between the hepatocytes and that fluid. All right, sitting in that parasinusoidal space is a really important population of cells. In a healthy liver, they tend to be relatively quiescent, meaning they're not really doing very much. Their main role is to be a reservoir or storage structure for some of our vitamin A, which is a lipid-soluble vitamin. And we can see here this healthy liver. These are the hepatocytes. Here we have our endothelial cells. Down here, this would be the blood flowing through the sinusoid. And this stellate cell, or Ito cell, is actually sitting in this parasinusoidal space. So these hepatic stellate cells, stellate meaning the shape of them, they're sort of star-shaped, will become active when there is some stress coming in from the hepatocytes, or to the hepatocytes. They become activated fibroblasts, and they start laying down collagen fibers in that parasinusoidal space. They also will help to sort of connect into each other, and they will condense that space, so it pulls together the endothelial cells and creates much smaller gaps. The significance of this is when the hepatocytes become stressed, so whether by increased toxin or viral infections and things like that, we're going to find that we want to close down the fluid movement through this space. And so we're going to lose the gaps here, which are called fenestrae, and they become much smaller. And we're going to make this a really thick area of collagen, which makes it very difficult for fluid to diffuse in and out and around through those collagen fibers. And so that helps to protect those hepatocytes from further damage and injury, but does result in what we call liver fibrosis. So the fibrotic pathway is initiated by damage to the hepatocytes and the sort of activation of these Ito cells, which are mesenchymal cells, are going to sit and sort of just be waiting. They're connective tissue stem cells, and they get stimulated to become fibroblasts by the hepatocyte injury. So this is just showing you kind of a nice transition between what happens in a normal liver to what happens with liver injury and liver fibrosis. So again, these are the hepatocytes. I really like this drawing because it shows the microvilli of the hepatocytes. So this increases the surface area for movement of nutrients across the cell surface of these hepatocytes. It shows you the really nice space of Dicey, or Dis, where we have our Ito cell, or our stellate cell. We have our Kupffer cell sitting in the sinusoid. And so fluid from the blood or plasma would then travel into this space and get metabolized, the nutrients, and detoxify, all that good stuff. When we have injury, you'll notice that the microvilli decrease. We're going to see that our gaps or spaces are going to get smaller, and we're going to have lots of activated Ito cells starting to lay down collagen fibers. And so this is going to start to harden the liver, make it less flexible, make it less likely that nutrients get exchanged and metabolized and detoxifying happens. And so this is the beginning of the pathology for liver fibrosis. All right, I just want to talk a teeny bit more about fluid movement through the liver. Again, remember, we're going to see blood moves into the liver through two entry points: the hepatic artery, which we have about 20-25% of our cardiac output, bring in oxygen-rich blood. We have our huge volume of blood traveling in from our GI tract, carrying our workload blood. As this blood enters, you're going to see here that essentially these two blood vessels will merge into the sinusoid. And the sinusoid will carry blood from the periphery of a lobule past the hepatocytes, past their parasinusoidal spaces, and toward the central vein. So blood vessels will branch into smaller ones, supplying each of these lobules. Blood is always going to enter at the periphery. So periphery meaning this outside edge. And if you think about, again, a bike wheel, this would be where the tire is around the outside edge. And blood is then going to basically travel through these spaces between the columns of hepatocytes through the extensions of these little blood vessels, which are called sinusoids, to the center, which is the central vein. So traveling down through these sinusoids, passing all the hepatocytes, exchanging in that parasinusoidal space with the hepatocytes, and eventually processed blood is going to then exit each of the liver lobules by a central vein. All right, so I have your sinusoids. This is basically the workload conveyor belt. It's carrying essentially blood down past all the hepatocytes. Remember, the fluid part of the blood, which we call plasma, will sort of exit through the little gaps and spaces within the sinusoid endothelial cells and into that parasinusoidal space. So that's the space between the sinusoid and the hepatocytes, where we're then going to do all this processing. So then processed blood will then drain the liver lobule through that central vein. So in this artistic rendering, we would have blood flowing from the periphery down toward the center through all of these 6 channels. All right, so all the central veins unite into hepatic vein, and then we have 2 major hepatic veins from the 2 main liver lobes that then unite and will flow into the inferior vena cava and then back to the heart. So again, you can see that each liver lobule has a central vein, and these will then all unite into a right and left hepatic vein that then travels back to the heart. Okay, so last but not least is the bile canaliculi. Canaliculi is nothing new. You heard canaliculi when we talked about osteons. So the canaliculi were the little tiny canals through bone matrix that allowed exchange to take place. Here the canaliculi are going to be channels through which bile produced by the hepatocytes is going to drain. So remember, bile is a waste product. It just happens to be a very important waste product in its conjunction with digestion. So as red cells are broken down and bile needs to be released from the body, it's actually excreted through the digestive pathway. So that's its exit route. And so hepatocytes are going to make this product. Bile will move counter current to blood. So it's going to move from the center of the lobule to the periphery. So you'll notice here the arrows for bile are going to be just opposite to the arrows for blood. So bile is going to be produced by these cells. And it's going to be moving toward the outer edge of the lobule, toward the periphery. And when we look at the canaliculi, they're really just gaps between the columns of hepatocytes. So they're sort of on the lateral sides of the hepatocytes that kind of have this indentation with microvilli into them, where bile is kind of deposited, and then it sort of flows between those hepatocytes. All right, so this is a scanning electron micrograph. This one's not colored, but we can see here a blood cell. And so this space here, this is a sinusoid. Down here, this is another sinusoid with a channel going down into the lobule there. These are the hepatocytes. So we're looking at the interior of the hepatocyte. And this tiny little space, that's the bile canaliculi. So you have the canaliculi. So remember, between the hepatocyte and the sinusoid is the parasinusoidal space. So I can see this space right here that kind of looks really fringed. This is that parasinusoidal space. You can kind of see it looks like it has a little bit of fringing on it. So that's all the microvilli at the surface of these hepatocytes. So that's the parasinusoidal space. On that column, here, of course, is the parasinusoidal space between the endothelial cell of the sinusoid and then at the surface of the hepatocyte. So here's another good view. This is parasinusoidal space up on the top up here. Okay. All right. So bile flows away from the central vein toward the periphery, and eventually all the little bile ductules will collect into a larger bile duct that eventually will leave the right and left liver lobes. So we'll have a right hepatic duct and a left hepatic duct that will be carrying bile. Okay, and this is again just another nice artistic rendering. Here we see the hepatic portal vein carrying in basically a huge workload of raw nutrients to be processed. Here's the hepatic artery bringing in oxygen-rich blood to these columns of hepatocytes. We can see some Kupffer cells. These are nice immune cells that will begin phagocytosis sitting in the sinusoids that help to kind of turn over some senescent red cells or any cellular debris. And then we see here the space of Disse, or the parasinusoidal space, and the sort of aqua color cell. That's one of our Edo cells or our stellate cells that are quiescent at this point, hopefully, and just being a reservoir of vitamin A, but can sort of increase sort of the viscosity of the space of Disse, so to regulate how quickly fluid moves through. And then lastly, we see in green here, this is the bile canaliculi that would be carrying bile from the center of the lobule out toward the periphery. All right, and then this is just another artistic coloring to a scanning electron micrograph. And if we're just numbering all these different structures, number 1 would be the bile canaliculi. Number 2 is the sinusoid with a nice blood cell in it. It's a red blood cell. Number 3 is one of the Kupffer cells sitting in a sinusoid. Number 4 is sort of a column of hepatocytes. You're going to see that they have gap junctions, so their cell membranes are relatively fused with each other one by one, so it's hard to see as they are navigating through a column. And then number 5 is the parasinusoidal space. So that's parasinusoidal space here, and we can see a little bit of the parasinusoidal space here. Again, we can see parasinusoidal space up in here. Okay, and then almost last but not least, we have what we call the portal triad or the hepatic triad. So at the 6-sided lobular structures, at the points around it, we're going to have 6 points where we have the portal triads located. And so this would be the hepatic portal vein, the hepatic artery, and the bile structure. So if we're looking, you can see at any one of these points, you're going to see a 1, 2, 3 organization. 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. In fact, a little video, that's one of the things that they mentioned when you're trying to view liver histology. You want to look for the central vein, and then you want to outline the 6 hepatic or portal triads. All right, we talked a little bit about bile drainage in lab, so lab 15, and I wanted just to re-mention that. We have 2 major ducts draining the right and left liver lobes. So we have the right hepatic duct and the left hepatic duct. That's what we're looking at up here. See if I can, there you go. Up at the top here, this is the left and the right hepatic duct. Anytime you have 2 ducts combining, they form what's called the common. And so we have the common hepatic duct that will essentially be draining bile produced by the liver. The common hepatic duct will then merge into what we call the common bile duct. The common bile duct is named common because it's going to be collecting bile from both the gallbladder as well as the liver. So we have 2 sources of bile that will be moving into the common bile duct. The common bile duct will travel down and then into the head of the pancreas, where it will join with the pancreatic duct to then enter into the medial border of the duodenum at the hepatopancreatic ampulla. Again, there's a little ring of smooth muscle called the sphincter of Oddi, or the hepatopancreatic sphincter. The papilla just is this little nub or sort of remember cone where we're going to see the entry point. So last but not least is the cystic duct, and the cystic duct is sort of bidirectional. So when bile is made, it's made all the time. We're not always releasing it. So the sphincter of Oddi is going to be closing off the entry into the small intestine. And so bile then starts to back up as it's being produced. And if we don't release it, that bile then is going to flow into the sort of path of least resistance. And so it's going to travel into the gallbladder. The way it gets into the gallbladder is through this duct called the cystic duct. The cystic duct is not only a way into the gallbladder, but also a way out of the gallbladder. So the cystic duct is going to drain and fill the gallbladder. The common hepatic duct is going to drain the liver. And then the common bile duct is going to drain the gallbladder and the liver into the duodenum. All right, so I have the right and left hepatic ducts are going to collect bile from the right and left liver lobes. So in this drawing here, they've kind of cut the liver, and you can see the left hepatic duct and the right hepatic duct uniting into the common bile duct. And then we see the gallbladder sitting under the right liver lobes. It's the cystic duct. And this is the common bile duct that would then enter into the duodenum. I also have, again, the cystic duct. This is going to be this bidirectional duct through which bile can fill the gallbladder as well as drain the gallbladder. And then again, the common hepatic duct, which is referring to the fact that we have bile from two separate sources that travel through that common bile duct. So we have bile coming from the gallbladder as well as bile coming from the liver. All right, again, remember the common bile duct is going to unite with the pancreatic duct in what we refer to as the hepatopancreatic ampulla, and that's going to drain into the duodenum. And we're going to find that, again, the common bile duct and the pancreatic duct are going to have that common entrance. Okay, so this is a drawing. Obviously, here we're looking at the muscularis externa, the inner circular and longitudinal of the small intestine. And all this smooth muscle here is what we refer to as the sphincter of Oddi. It can regulate the release of bile and pancreatic juice when the stomach is distended and an individual's chewing. We saw the quick little video of bile being released, but this is just looking at, again, this common bile duct. We can see we're on the inside of the small intestine here. So these are plicae circularis, and they're trying to show the little papilla, which is this little kind of cone here. And if we were to kind of go into that, this is the sphincter of Oddi here, or hepatopancreatic sphincter, that then would enter into the ampulla. Okay, and then last but not least, I just want to talk a little bit about liver cirrhosis or scarring of the liver. Initially, it begins as scarring coming in from the laying down of excess collagen fibers, which we refer to as fibrosis. And this is associated with the destruction of sort of normal liver architecture or hepatocyte structuring. Most common cause of this is going to be alcoholism. So taking in the toxin of alcohol can damage the hepatocytes. Or it can be a viral infection like hepatitis B or C. So here we're sort of looking at normal liver, and then this would be a liver where we have liver cells getting damaged, and those mesenchymal cells will differentiate and start to lay down collagen fibers, and then also can differentiate, and we're going to see an excess of adipose forming within the liver itself, so forming fatty liver and fibrotic liver. So this is a quick little video looking at liver fibrosis, and then we're going to finish off looking at just one of the major problems with fibrosis.
Liver fibrosis is scar tissue in the liver, and this typically happens after you have been affected by a liver condition that lasts for many years or decades. Progressive fibrosis can lead to cirrhosis of the liver, which put people at risk to develop liver cancer or require a liver transplantation because the liver is no longer functioning well. The risk factors for liver fibrosis are mostly chronic conditions that affect the liver. Some of these are very well known, such as too much alcohol or chronic hepatitis C virus or chronic hepatitis B virus, and increasingly in the US also non-alcoholic fatty liver disease, which we see in people with diabetes and with obesity. The CADC is an excellent center, which, because of the infrastructure and the efficient organization, allows us to use state-of-the-art diagnostics and therapeutics, which sets us apart from other liver centers in the city and the country.
Okay, and so this is just looking at sort of the progression that happens with liver fibrosis and then eventually cirrhosis, where we start to see deposits of excess adipose or fat. And normally we'd see blood flowing in through the hepatic portal vein and the hepatic artery. But as we start to see progressive fibrosis and fatty liver deposits developing, it kind of stiffens the liver and it makes it pressurized, and it's much harder for blood to flow into the liver. We'll see that the sinusoids start to narrow their gaps, and it makes it more difficult for metabolism of nutrients to occur. And as this continues, we're going to find that there's so much back pressure, it's really hard to push fluid through the liver. And remember, all of the digestive tract has to travel through the liver, that when it becomes really fibrotic and it becomes very difficult for blood to flow in, you're going to sort of get this back pressure that causes not just the liver, but all the abdominal organs to start to seep fluid. Which is what we're looking at here. So this is all this seeping of serous fluid. When that happens, all that fluid starts to accumulate in the abdominal cavity. And if you see someone with end-stage liver failure, they often look like they have a very distended abdomen, almost like they're pregnant. And what that is fluid called ascites. It's this peritoneal fluid that is accumulating in the abdominal cavity and causing that abdominal distension. Now, the most common cause of death associated with liver failure isn't actually the problem with the liver not being able to detoxify or metabolize, but the most common cause of death with those who have liver fibrosis and cirrhosis is in fact a esophageal hemorrhage. Because what happens is that blood that's supposed to go into the liver here can't get into the liver, and so that blood has to find its way back to the heart. And so it's going to take kind of an off-ramp through the esophageal vessels, which are not supposed to handle that volume of blood flow. It's kind of like if you're traveling from San Francisco to LA and I-5 is sort of closed, and you're like, oh, shoot, how do I get around it? You'd have to get off on one of those side streets. And if you tried to push all of the I-5 traffic through one of those little tiny one-lane side streets, it's just going to overflow and back up. And you're going to see, actually, this happened. I can't even remember when it was when I was driving down. And I can't remember why I-5 was closed. And they had all these people driving not just off the streets, but off-roading it through fields and stuff. It was crazy. And when it happens in your body, these esophageal vessels can actually rupture. And so you almost always die of internal bleeding. And so this is just a few statistics. And on average, when we look at prevalence in the United States of liver cirrhosis, they estimate that about 400,000 individuals a year are diagnosed with liver cirrhosis. That results in about 375,000 hospitalizations every year and about 25,000 deaths in the United States annually. The mortality rate is about 85% within 5 years once the liver starts to decompensate. So that's a pretty high level of mortality associated with liver failure. So important to make sure you keep your liver healthy. All right.